My name is Dr. William McCoy. I am the director of the Rutland Institute for Ethics. I am so happy to see all of you here. I know we have others that have joined us online for a very dynamic topic on tonight. And it is so good to see all of you and the interest that you have in the topic. We are encouraging you to write down any questions that you have. For those of you that are accessing it online, you can always write a question in the chat as well. But we will have time for Q&A at the end of this. I want you to get as much out of this as you can. This is a whole new setup for us. Please, if we have some technological issues, which we actually did in our last session, I think we worked through them, but please be patient with us if we hit a snag. As you well know, technology does not always like to cooperate with you. At this time, we're going to turn it over into the hands of Dr. Tennyson, and he will introduce our speaker.
Well, good evening, everyone. Thank you very much for coming to the talk. This is going to be an exceptionally exciting talk, I think, with a lot of very cool insights into best practices in scientific research moving forward. So it's my pleasure to introduce Professor Schrag, who did his MD at Loma Linda, and then he did his residency at Yale, and he is currently an assistant professor in the Division of Neurology at the Vanderbilt University Medical Center.
Well, now, if it's not that exciting, okay, I'll hold it against me. There's more of a buildup than I can deliver for you. I'm actually a little bit impressed to see you all come out, because usually when we tell people we're going to talk about research integrity, they assume the whole lecture is going to be cheating's bad, don't do it, and then you can all go home and go have dinner. More interesting than that. And I'm going to talk to you very specifically about how these issues have affected Alzheimer's disease, but I hope regardless of what discipline you all are working on, you'll find something here that is of interest. And certainly there's some realities facing us as scientists about how to deal with the enlarging literature that we're interacting with and figuring out how to make this reproducibility crisis, right, that we struggle with, how to solve it. So let me just start by telling you a little bit about it, if I can get this to cycle. First slide is always a challenge. Come on. There it goes. All right. I study a process called cerebral amyloid angiopathy. We don't think of Alzheimer's disease as being a blood vessel disease, but for a lot of patients it is. And that variable, that deposition of beta amyloid in the vasculature in the brain, in some patients with Alzheimer's disease, is a really tough problem to solve. And it's the source of some of the side effects we see in some of the experimental drugs. So we're trying to understand why the blood vessels fall apart. My research uses advanced microscopy and image analysis techniques. And some of those things we're going to drag out of the traditional scientific setting and try to apply them to some forensics. And so the reason I'm here is because occasionally I consult in the research integrity space, and it's that second hat that I wear that we're going to mostly talk about today. But I want to show you some of these advanced 3D microscopy techniques that we use. And one of the things that we're trying to understand is how a blood vessel in the brain, which is supposed to be nice and straight and smooth, could turn into something that looks like this, all twisted with areas that are dilated and areas that are constricted, and what the consequences are when that happens. And it helps if you're able to see that blood vessel in 3 dimensions under a microscope and get, a better understanding of the relationship between different markers. And so we like to use these nifty tools like light sheet microscopy and clarity to try to understand how these changes in blood vessels occur. I have an incredibly talented staff scientist named Lisa Ventura-Tunes who puts these videos together for us. And from these 3D renderings, we can make measurements of the relationships between different molecules. And it's really an outstanding platform for trying to understand the human brain. So let's talk about research integrity and what goes on when you do image analysis forensics, right? Which initially seems a lot more boring than all of that, but surprisingly down at the core, some of the things are the same. Now, I showed a few of you guys this picture earlier, but I want to just start by seeing if we can pick out of the audience the people who have good innate skills in pattern recognition. So I'm going to start by telling you that these two images are both very heavily manipulated. And I want to see if anyone can pick out what's wrong with them. So these are pictures from two different rabbits. One's supposed to be a normal rabbit brain. The other is supposed to be a model of Alzheimer's disease. And the arrows that you see were in the published version of the image, and they pointed deposits of ferritin. The idea was that iron accumulates in these brains and drives some reactive oxygen species generation and so on. Now, I know a few of you have seen this before, and I'm not going to quiz anybody to see if they can pick up on what's wrong with this image. But the problem with an image like this is often it's pretty subtle things that initially tip you off that maybe something's wrong. And then the more you look and the more you look, the more you start to see that most of what you're looking at is fantasy. In fact, I'm not sure this particular experiment ever even happened. Let me show you the first tip-off. First tip-off's right there. You see what I'm pointing the arrow at? There's two little notches in the image, one right at the bottom margin, a little notch that just doesn't look like it belongs there. And there's a similar one in the middle, just a square off corner of that image. You see it? You believe me? Here, I'll make it bigger. You see that little corner? Right? There's not much in the brain that forms geometric shapes like that. There's a bit of a tip off, right? That most things in the brain don't have corners. Right? So it's not always that obvious, but that's a helpful place to start. But that's not where we finished with this guy. Now, I want you to look at this. Okay? Everything that I've got marked out has been introduced into that image with a clone stamp tool in Photoshop or something like it, right? So each of those little red boxed structures are the same. And if you magnify them, unfortunately, the author has gone ahead and magnified it for you, so you can see all those interior details with a great deal of detail. You'll see that all of those fine details are duplicated, and the orientation is the same across. Oh, not just the far image, also down here in the corner. And also, this entire corner of this image seems to be the same as the entire corner of that image up there, except for the photoshopped in neuron. And then I'll point you over here to these two things circled in red. You see these two structures? Look how close these two are. I put them up here slowly. See up here. The more you keep looking at these images, the worse this gets, right? This is big league bad, right? You don't want your name touched in this paper. Don't look to the names too closely, okay, guys? Okay. All right, so let me tell you a couple of things, okay? First, let's start with a myth, something that you've probably been told a long time. If you haven't been told, this has been implied to you, and that's that the manipulation of scientific data is rare. And I want to start by dispensing with that myth. Data manipulation is not rare. Unfortunately, I don't think it's hugely common, right? But it happens enough that you can't ignore it, right? We can't ignore it. The consequences of ignoring data integrity concerns are pretty substantial now. So we have to take this seriously. A relatively small portion of data manipulation happens in avenues where we can forensically analyze it, right? When you look at a typical paper, a lot of the data is contained in tables and things where you just have to take people's word for it. And so when you see us talking about Western blots and microscopy, a big portion of the reason why we're talking about it in those settings is because we can access some original data to apply some forensic tools, okay? I don't think it means that that's the only domains where these problems happen, right? It's an area where we have access. And then I think most of you who've been doing this business very long know that there's an alarming number of scientific results that cannot be replicated, and that's a little bit troubling. There are certainly good, innocent reasons for why a piece of data might not replicate, but one of the reasons that does crop up is scientific misconduct. And it should be okay to say that out loud, right? This is a complex human endeavor, and anytime you've got tens of thousands, hundreds of thousands, millions of people working in an area, there's going to be a certain amount of error and there's going to be a certain amount of mischief. And it's okay to say that. And we need to develop systems that are resilient to that, that have ways of handling a certain amount of problems in the data. And we need to think about what are we going to do, not if, when you read and handle data that is not entirely reliable. How are you going to react to that? How are you going to not get taken in by that? So one of the problems that we have on the consulting side is that when I take an image like that to a journal editor, all too often they say, yeah, it could have happened, right? And you're like, nah, that couldn't have happened, right? But this is not a productive conversation, right? And so one of the things that has to happen is you have to prove your case, right? You have to really try to take what you're seeing and apply analytical tools to say, okay, no, what actually is the probability that that happened by random chance, right? And it turns out we have tools from the image analysis side of this to do that. A lot of things that we do in analyzing microscopy images or any other image is to ask the question, what is the probability that these two things have these results by random chance, right? We're good at that kind of stuff. So you can pull those tools into this space. So the first part is to prove that in fact it's duplication or some other manipulation. The second is to create the argument that that manipulation is inappropriate. Sometimes it's not actually. And then 3, that it alters the underlying meaning. And then 4, that it wasn't merely an error. Okay, let me give you an example. So this is a relatively simple one. This is a paper that came to a journal club that we were having with our students recently. And I'm going to tell you that those 2 boxed images overlap. That's not immediately obvious, but if you take a look at it for a minute, you might see the overlap. Is it jumping out at anyone? How do those 2 images look? I'm going to make it a little bit easier. I'm going to flip one green, leave the other red, and I'm going to stack them on top of each other. See it? So I think it's pretty obvious, and probably you could go to a journal editor with that image and say, yeah, this one will probably fly, right? But let me show you how we might analyze something like that. Okay, what we're going to do, we're going to do the same sort of thing you would do with co-localization analysis in your microscopy, right? We flip them to different colors, and we're going to ask, when you go pixel by pixel through that image, to what degree do the 2 signals correlate, right? So we're going to convert them to grayscale, we're going to colorize them, we're going to try to reverse any manipulations that we think have been applied. So if it's been stretched, we're going to unstretch it. If the curves have been adjusted, we're going to try to unadjust them. We're going to try to put it back so that the 2 images are as close to what we think the original is as we can get them, okay? So anything you think was done to that image, you can reverse engineer and try to put back. And then we're going to isolate the overlapping areas, and we're going to ask that question, what happens when you plot it pixel by pixel by pixel, okay? So I've shown you here the aligned panel, taken out the pieces that are not going to behave. And I've shown next to it a random alignment, basically the alignment that was published, right? To show you what these 2 scatter plots look like, right? And what you see in the top panel is the alignment of the 2 images that have been rotated. And you can get a quantitative output that says you've got an R² value of 0.92. There's no way that 2 microscopy images by random chance should give you an R² value of 0.92, right? That just doesn't compute. When you look at a random one here, you get an R² value of 0.088, right? So now you can put some numbers around what the probability is, and you can take those distributions and actually calculate it out for all the lawyers in the audit, tell them the odds of this are 1 in whatever, it's a very big number, right? More than a billion, more than a trillion, more than whatever, 100 trillion, right? It's not very likely. You can do the same thing with the Western blot. So here's a Western blot, not a particularly good Western blot. Lots of problems with this Western blot. And if anyone's really clever and says there's other things that overlap aside from what we boxed out, you're right. There's lots going on here. But you can see the 2 boxed areas look surprisingly similar. And I'll tell you something that we really appreciate in this business, and that is the detail in the background, right? When you look at the background of these images, the background of a Western blot should be approximately random, right? So now if you can get an alignment out of stuff that should be random, you can really use that to interpret what you're doing. But the actual bands of the Western blot might look the same for a good reason, right? You ran them from very similar samples at the same time on the same equipment, right? There may be very good reasons for the bands to look the same. And so you have to interpret these numbers within the context. But when you're looking at the background, you're expecting randomness. You can get good signal alignment. You've got a strong case to make. And here we hit something close to a record, R² value of 0.990. We see how basically everything when you merge the 2 images stays yellow, right? So anything that was different between the 2 of them would either be red or green. Let me show you what that looks like. So you can actually use this to show elements that have been introduced into the image too. So let me show you this 2 different versions of what I thought were probably the same image published in 2 different papers. Okay, so these are this image over here in black and white, and that image over there in pink and white. Okay, they look to me superficially to be the same. You see a lot of things in the background that overlap. But there's also some things that are not shared. You can get this sense of what might have been introduced in these images. So we're going to go through the same process. I'm going to assign them to a color, and I'm going to overlap them. And anything that overlaps is going to convert from green and red to yellow. Anything that's shared turns yellow. Okay? Anything that's been introduced or taken away is going to retain its original color. Now, here's the important thing. Obviously, there's a few dots that look like they've been added in one version and one dot that's been added in another version. But one of the questions that we like to ask is, okay, what does it mean? Why did this happen? Why did somebody bother to put a couple extra dots in here? Right? And so this is where you have to have some expertise in this space to really decide, okay, does this actually matter? Right? What actually happens here? This is a model of Alzheimer's disease in rabbits. Right? In this model, you feed cholesterol to these rabbits, and over a period of time, they get plaques in their brain, the same beta amyloid plaques we see in humans. Right? The problem with this is a few years after this paper, these 2 papers were published, another group found that this was not a hugely reliable model and that a lot of the rabbits didn't get that many plaques. And now this model's been more or less abandoned, right? But you can see why somebody who was working with this model, who didn't want to confront the fact that it's not a great model, or whose funding was contingent on that, but who thought they were going to lose their job if they didn't get papers for it, might say, oh, I'll throw in a couple extra plaques to make this thing look a little better, right? So the context, the scientific context is very important to try to understand why these things happen. Okay. So why are we talking about plots and microscopic images when we're going to talk about Alzheimer's disease as well? I could tell you quite a few stories now because the landscape of Alzheimer's disease is littered with bad stories. In the last couple of years, we've had a number of high-profile collapses on biotech companies whose stock prices cratered because their scientists had these sorts of problems in their preliminary data. Athira Pharma's CEO's entire dissertation was filled with this sort of thing, and she was forced to resign and the stock price plummeted. Cassava Science is going through very similar things. But I'm going to tell you a slightly different story. I'm going to tell you about the amyloid hypothesis in Alzheimer's disease and how these sorts of problems might have gotten influenced the way we actually formulate this disease and how we're targeting this as a field at large, not just one company, not just one lab, not just one scientist, but how we as an entire field have kind of gone down what might be a rapid trade. So Alzheimer's disease is a big deal. And I don't really think I have to make this point anymore. I probably didn't have to back when I made this slide either, right? But there's a lot of faces that come to mind when we think about Alzheimer's. We think about our scientific leaders, we think about our political leaders, our sports figures, our actors, we think about our parents and grandparents, right? There's very few families now that have not been personally affected by this disease. But before all of these faces came along, there was this one shadowy face, right? This is the first patient that Dr. Alzheimer's met who had the disease that would go on to be named after him. And her name was Auguste Dieter. She was a relatively young person for a diagnosis of Alzheimer's disease. And he described his interactions with her, that she was having a hard time speaking to him, that she was having a hard time following the track of a conversation, that she would forget the words for common things, that she couldn't tell you the names for things, that she would repeat the same answers to questions even though you asked a different question, right? These are all things we might have seen from family members affected by this disease or other people that we know. And when Dr. Alzheimer's looked at her brain, he found a couple of things. One is he drew pictures of these tangles in the brain, right? We now know a protein called tau that gets phosphorylated and twisted up. And this is a protein that's essential for the structure of that long, delicate axon, which is how a neuron conducts its electrical signals and creates a network in the brain that allows you to think and interact with the world. And he also found beta amyloid plaques. And at the time, of course, we didn't know that they were made of beta amyloid, but he saw these little structures here. And this is a slightly more modern version of what he would have done to stain them. But essentially, these little spots here, I've drawn a large arrow to one down here, these little red spots, are clumps of proteins that are forming outside of the neurons. and they're causing toxicity to the adjacent axons, right? So each of these little green blobs around that plaque is an axon that's got stretched out and is no longer behaving normally. So these tangles and these plaques have become the hallmarks of this disease. And in the early 90s, there was this initial presentation of a grand hypothesis to explain why Alzheimer's disease happens. And the hypothesis was it's the plaques, right? The plaques are the first problem that come and initiate all the other things that we see. It initiates the formation of the tangles, the neurodegeneration, the loss of bulk from the brain. Eventually, the brain shrivels up. Right. The thought was that the very first step in that chain of events was beta amyloid plaques. Interestingly, at the same time this grand article was being published in Science, there was a paper being retracted in Nature from the same author. Right, from one of the authors of this grand amyloid hypothesis. They had been trying to build a mouse model of this disease. One of the first efforts that was thought to be successful of modeling Alzheimer's disease had been published a few months before in Nature, and it got a lot of press. And you know that the retraction is bad when they put it as a headline in the Washington Post. So this is not a good way to get your retraction out there. But what they found was that this brain tissue that they thought looked a lot like the human version of Alzheimer's disease looked a little too much like the human version of Alzheimer's disease. In fact, it was human tissue that they were staining and publishing. And so that study had to get retracted. And there was a series of years where we struggled as a field because we didn't have a good model of the disease. And that really helped when you're trying to develop treatments. So here's a look at these. You can see that we've still to this day not really got mice to develop these tangle structures like you see in C and D, right? So it's still pretty clearly something that we would struggle to produce an image like that from a mouse today. But instead, a few years later, we got a better mouse, and this one has become a staple. And so a scientist named Karen Ash, who became fairly famous for working with Stan Prusner, who won the Nobel Prize for studying prion diseases. She cut her teeth studying the genetics of prion diseases. She struck out on her own and she built this mouse, modeling it on a number of mutations that cause Alzheimer's disease very rarely in people, this early onset version that affects well less than 1% of patients. She put those mutations in this little mouse and they get Alzheimer's disease or something that looks like it within a year or so. This was a major breakthrough. Now we've got an animal that we can study in behavioral tests that we can give drugs to, we can study their brains. This is a huge, huge step forward. In fact, she was part of a race to develop one of these animals, and a drug company developed a very similar animal around the same time. And they used it for developing a vaccine against beta amyloid, which is an incredibly clever way of solving this problem. If the plaques are the problem, why not wake up somebody's immune system and see if you can get beta amyloid out of the brain, get rid of it, and hopefully people get better. And the good news is it worked in the mice, right? It turns out we've done that over and over and over again in the field, right? We can really cure these mice, right? But in people, it's a little bit more complicated. People tend to be complicated. And the clinical trial was a little bit disappointing for two reasons. One is that the patients who were treated didn't do as well as everyone had hoped. There was not a real clear improvement in their clinical trajectory. But the other problem is they started to develop swelling of the brain. They had a side effect from this, which is not entirely unexpected when you initiate an immune reaction against the brain. But something like 8–9% of these patients developed encephalitis. And so that effort was suspended. But the underlying idea got a lot of traction. And could you control this reaction and give people antibodies or some other trigger for their immune system to get rid of these plaques? And could you get it balanced right so that the side effects are under control and help these patients? And that's been the leading approach to treating Alzheimer's disease since this time, right? So there have been a series of many clinical trials trying to get the right antibody, just the right set of circumstances to make this work. So after this happened, this was probably the first big stumble for the amyloid hypothesis. And in the early 2000s, a lot of people for the first time were saying out loud what some people have been thinking for some time, which is maybe this is a little bit too simplistic. It is a chronic, lifelong disease. What are the odds that there's one and only one driver of this, that you can pull out this magic bullet and everything's going to be fine? Like, it sort of strains credulity. At least to me, it does. And certainly to large swaths of the field, it didn't. But there was this moment where the amyloid hypothesis sort of stumbled. And there was a bit of a reformulation time. One of the thoughts was maybe the problem is not exactly the plaques, right? But if you can get huge clumps of beta amyloid in the brain of these plaques, so it starts out as one little thread of a protein, maybe somewhere in between that spectrum, there's something that's selectively toxic, something that's special, that is the driver of Alzheimer's disease, right? So we call these mid-range clumps, these littler clumps, oligomers, right? And the oligomer hypothesis came along around this time. And the question was, could you specifically target not all the plaques that are causing all the side effects, but could you just pull out the really toxic forms and protect patients? And Karen Ash, who invented this mouse, and her all-star postdoc thought they found the culprit in a protein they named A-beta star 56. This was the silver bullet in the mid-2000s that was causing Alzheimer's disease. And they published this thing in Nature in 2003. And it's gone on to be one of the most cited papers of all time in the field of Alzheimer's disease, thousands and thousands of citations. And again, the concept was, could you leave most of this alone, just pull out that one clump? And I think even from the beginning, there was some skepticism that this was the only clump that mattered. And so I think from the outset, this notion that this was a silver bullet was questioned. And I'll tell you, I'm going to build this story around this paper falling down. But of course, no one hypothesis hinges on a single paper. So there is a context around this that's very important. There's lots of other studies, but this is a big one. This is part of the foundation of this notion of the amyloid hypothesis. And that went on to become one of the most cited articles in the field. And one trial targeting it after another, after another, after another, after another, they all failed, right? All of these antibodies going after slightly different versions of this protein that failed and continue to fail. And we keep asking, why do these drugs fail? So maybe the question is, we have to look at our assumptions, right? So we go back to this paper and we look a little bit more closely. So around this time, I was running a research integrity investigation on a drug company called Cassava Sciences. And I was trying to develop some of these techniques for quantitatively analyzing these images. And I was looking for other sources of images to run through this algorithm for analysis. And Sylvain Lesnie, this all-star postdoctoral fellow, his papers looked an awful lot like some of the stuff we were seeing from Cassava Sciences. And so simply because it was around and available, we were studying some of his work. And what we find is that when we find one problem in somebody's CV, we often find a lot of others. So that we sort of backtracked into this study. And I was being followed around by a reporter at the time. And so I wasn't able to do this as surreptitiously as we usually do. And I think they recognized that this was a big, big problem. And so together we analyzed this study, and I want to show you how we would approach going from something like suspicious to something that's seriously, seriously wrong. I think when you look at the coverage of what happened, you'll get a lot of people saying either this is the worst thing that happened to Alzheimer's disease and billions of dollars was wasted in this and that, or they'll say this had no relevance to this problem at all. And neither of those two things is true. This is important. This is a big part of the foundation of the hypothesis. But I want to show you that it's not a matter of one person's opinion versus another. I want to show you the depth that we can take this to. Okay? So here's one of the things that struck me. This is a Western blot looking at oligomers in the brain, right? So this is a stain for a protein called the amyloid precursor protein, which is this big black smear at the top, right? This is the parent protein, and then it gets clipped into a little tiny 4 kilodalton peptide, which lives way down here at the bottom. You see this little band down here. And as it clumps together, you get this little ladder on the way up. So when there's 2 of them, they're the next band up. When there's 3, when there's 4, when there's 6, when there's 8, and so on, they get heavier, and you see this ladder. And so this group is interested in the order in which these things appear as their mice get older. And they're interested in specifically this little 12 mark. See that? That's the A beta star 56 that they think is responsible for memory problems. And what they did with these mice is that they isolated this little 12 or this A beta star 56, and they injected it into rats, and the rats couldn't remember anything for a few days. Okay. So making sure that they isolated the right thing, the thing that actually said, ends up being a really important part of the process, right? So this is one of those control experiments showing that you're actually isolating what you think you're isolating. Okay. And what struck me is these two very different experiments looked more similar than I would have thought, right? So I boxed a couple of areas here that I think just look more alike than I would have thought. And if you look at them quickly, you would be right to say, no, there's a lot different between those two. But let me convince you. Remember I told you that the background is really important, right? In the background, right, you should be close to random, right? So I want you to ignore the really obvious bands for just a second. I want you to look at the air bubbles, right? If you take a bunch of little tiny air bubbles and you sprinkle them on a Western block, they should land in random places, right? So I think here I've got arrows to about 18 of them, something like that, in exactly the same spot on each of these membranes. What are the odds of that? No, I mean that seriously. We have some Mathew people here. Right? Can we actually calculate the odds of that? It's a few orders of magnitude less than 1 to a trillion, right? So when you do an analysis of a fingerprint, how many points do you look at? 12, right? So this is lower odds than your fingerprint matching, right? So the probability of these two things being derived from a different block with that pattern of artifacts in the background is exceedingly low. Okay. So what else? So what do we do from there? So then the next question is, if they came from the same membrane, how has it changed, right? What else has happened to it, right? One of the things you can do that you're not supposed to do to a Western blot is you can shift curves, right? It's okay to take and make it brighter or darker so long as you apply it to the entire membrane. But if you shift the curve so that some spectrum are affected more than the others, you're not supposed to do that, right? So these curving diagrams show you when you superimpose these on top of each other that these have not been adjusted in linear ways, right? So different sets of bands have been adjusted in non-linear ways, which can change the quantitation of that Western blot, right? And here's something else that's very interesting. A couple of things here. One is right down here. Well, let me start by what's at the top, right? So these are supposed to be different animals, right? So you see the age is listed out up top. You see those don't superimpose, right? So these are being described as animals of different ages. And as the animal goes from younger to older, you should see more of these larger molecular weight species. So you start to see the former appear. And you can see that you've got this one very green band, right? That turns up in the 13-month-old animal, but not the 9-month-old animal, right? So when it stays green like that, instead of turning yellow when you superimpose it, you know that it's been added or subtracted from one of the images, assuming they came from the same origin, right? So we've got a band that looks like it's been added to one of them. And the question is, where's the source of that band? Could you identify where that band came from? It turns out, to your eye, it looks an awful lot like the little yellow band right underneath it. In fact, if we put the two of them here and superimpose those, we get an R-squared value of 0.9936. That's a pretty good number. Okay, so it looks like not only can we say it's been introduced, but we also know the source of that band. We know where it came from. The question is, are there any others, right, in this thing? Are there any of the bands that have been duplicated? And I'm going to pull out this. So this was hard to do until Karen Ash shared with us some of the original images at high resolution values. So in order to do this well, you need really good quality images so that you've got a lot of detail, right? But the two areas that are boxed in red and green here, I've brought down for you that we're going to focus on, okay? Actually, we're going to do the black dotted box there, we'll pull them down. And I think just to your eye, these are complex shapes, right, that look the same on the two layers, right? So this is the same level where we saw the band added. So these are more bands in that space. And if you superimpose those, you get, again, an extremely high R-squared value. I've magnified one up here because I want to show you it's not just a blurry smudge, right? If you look at the internal texture of that band, right, there's a lot of information there. There's a lot of ups and downs. There's a lot of detail. Align every one of those pixels up is incredibly hard to do. So we thought it was good to have 16 or 18 air bubbles aligned, but this is way, way better. So we're left with a situation where there's very little doubt that these two experiments are not as described, that the source of these Western blots and presumably of the material that was ultimately injected into the rat. Who knows what it was, but it wasn't as described, right? The problem was we found these sorts of problems throughout a body of work from these co-authors. So in a fairly high profile paper in Brain a few years later, we saw this Western blot. And this Western blot appears to have a band that looks a little bit odd here. What we've done is we've just darkened everything so that you can see the background, and you can see this area around this last band where the 6-mer, it doesn't look right, right? Sometimes that's all forensics is. You just look at it, it doesn't look right, right? So we went to them and asked them, would they share with us the original blots? And it turns out it didn't look right because somebody photoshopped them in, right? So there's the original blot on the right. We've darkened them both so you can see the published version versus the not published version. And when we superimposed them, everything in red was added, everything in green was taken away from that Western blot. Believe it or not, the authors thought that was okay. The editors too. The editors published a correction, said no big deal. It turns out that sometimes you think that journal editors and regulators and these folks are going to be really cued into these things, and it's not always true. The system is not really designed to correct these sorts of problems, and so it's one of the reasons you have to be aware of them, is because it's not really in anybody's interest to fix this, except for moving your science forward. I'll show you this one. This one I thought was particularly impressive. I took this Western blot also from Dr. Lesney, but without Karen Ash's involvement. By the way, I don't think Karen Ash had anything to do with any of this, right? We've talked to her extensively. I've analyzed her papers that did not involve this particular postdoc and found no problems. Okay. We converted these sets of fluorescent Western blots to black and white the way we're used to looking at them, and we've blown up the background again so you can see it. I want to show you the number of copy and paste across the background here. You see how it goes all the way across the image 5 or 6 times. You see how those features repeat over and over and over again. Not ironically, I think this air bubble on the left looks a little bit like a middle finger. All told, by the time this was over, we had looked at 20 of Sylvain Leslie's papers that were in, that were showing serious signs of problems. And despite bringing these concerns to the NIH and to the University of Minnesota, Leslie was awarded 2 large research grants since the time. And to date, none of these papers have been retracted, not a single one. But it doesn't mean that the world hasn't taken notice. This has been profiled in more than 300 newspaper articles on the front page of the Times of London, and profiled in the Moth and the Wall Street Journal, USA Today. So everybody knows, right? And this is unfortunately what's going to happen if we don't regulate ourselves, is that the community is not going to tolerate this forever. Dr. Ash has acknowledged that these images are inappropriate, but she's convinced that this was purely cosmetic and didn't alter the underlying data, which remains to be seen. But I will say that they shared with us original data for this band, saying exactly the same thing, that it didn't alter the underlying data. And when you have the original and you have the published versions, you can actually put some of those claims to the test. So here, Sylvain Lesney is introducing a new way of purifying membrane-bound proteins. Okay? So he's got this old way that he's profiling here on the right, and then a new way that are the 3 bands in the middle. You follow the structure of this. He's trying to show you that the 3rd band is much more enriched than the 2 that precede it compared to these other 2 bands. So the notion is, if you want to get a membrane-bound protein, you take the soluble proteins away by pouring them out in solution with not a lot of detergent. Like, you know, to add detergent, you should bring the membranes down with it. That's the traditional way of this. And he's arguing that this new approach is more effective, right? And what you see here is about a 35-fold enrichment in his membrane fraction compared to the areas where you shouldn't see membrane-bound proteins, versus a 2.5-fold enrichment. Okay. When they sent us the real Western blot, this is what they showed, right? And again, the claim was that this didn't change, but it turns out we can actually measure these things and we can see how they change. So when you superimpose them, you see what's added, you see what's taken away, right? Everything in green's been taken away, everything in red's been added. And what was in lanes 1 and 2 was reduced by about 90%. What was in lane 4 was increased by about 500%. And the effect was to completely invert the interpretation of this. It turns out the traditional fashion method was much more efficient than the novel method of doing this. And so I think when you hear somebody dismiss this as, oh, it doesn't affect anything, I want you to hear this echoing in your ears. You couldn't trust them the first time. You maybe shouldn't trust them the second time either. Okay, so the outcomes. The oligomer hypothesis is a huge research priority, garnering a massive investment in research dollars. It doesn't just hinge on this paper, but this was a big part of the foundation of that story. 20 years of clinical trials targeting beta amyloid failed. In many cases, the drug did effectively lower beta amyloid. And the most recent trial, lecanemab, is sort of the one exception that just barely showed a clinical effect, but this was just barely measurable and probably not clinically meaningful. And it doesn't align with the hypothesis that says this is the sole driver of the disease, right? It may be a small symptomatic effect, but this isn't a disease driver. If you remove it, all you can see is this teeny tiny change. So the amyloid hypothesis is a deeply rooted dogma, but the fundamental assertion that beta amyloid is the primary driver of Alzheimer's disease is clearly wrong. I want to talk to you more about research integrity because I think it's more broadly applicable. Manipulation of scientific data is corrosive. It's corrosive to your work. It's corrosive to the culture in an academic community. It's also corrosive to public trust, right? Patients and families and people living out in the community don't need to hear many of these stories before they don't want to take your vaccine. Lapses need to be quickly resolved with very clear consequences. Publishers need a clearer role in how to handle this. I think that we've seen really, really sluggish response in case after case after case from publishers and journals. And there's a danger to focusing too much of the resources on a single idea. I think that one of the things that needs to happen as a field is that we need to fund ideas a little bit more broadly so that there's more competition in these spaces, because this drive for everybody to think the same way as a way of promoting data fabrication. Another thing that I think is worth telling you is that these things can be very personal, and at the other end of this story is usually not some bad guy, right? It's often a human who's trying to survive. I'm sure there are different pathologies at play, but this story that I told you at the beginning was done by somebody very important to me, and that's my name up on the authors list, right? So these were my very first papers, my first and second paper when this occurred. And Othman Greavy, who was the individual who's responsible for this, changed my life, right? I wasn't going to be a scientist before I wandered into his lab. I was certainly not interested in studying Alzheimer's disease at that point. He taught me a lot. He put me on a trajectory that was very different than what I was going to do. And yet, we cannot ask people to maintain the standard that we wouldn't hold ourselves to, right? And so this is a big problem. And when you see it as a co-author, you have to decide how you're going to respond. People get very worried about their reputations. People get very worried about what's going to happen to them. Don't. Just retract it. That's mine too, by the way. And last week we retracted this. It was hard. It took 9 months of haggling with this ridiculous journal to get them to track, which was clearly a fraudulent study, right? This is just a problem with journal after journal after journal, so it's not that easy. But I want, when you see these things, I want you to think through it before you get there, because there's a lot of ways of talking yourselves out of doing the right thing. But if you're a co-author, just retract it. But you know, Othman is still important to me, right? Still a very special human being who made some really bad decisions. And I bet you there's people who say the same thing about Sylvain Leslie and other people who've been pulled into this. In fact, I've heard it quite a few times today. I would never imagine it from that person, right? That's certainly how I reacted to Othman, right? Sort of a father figure to me. So, Othman, when I was a new faculty member at Vanderbilt, asked me to come and talk to the graduate students at the University of North Dakota, give them some advice, which I was in no position to give, having just started a lab, literally had no data, no experience that don't get many invitations at that stage of your career, so I went. And I gave a talk that day with Hoffman in the crowd, and I gave the 10 commandments for being successful in grad school. Commandment number 3 was pick good mentors. I've never seen Hoffman more drunk than he was that day. But the 10th commandment, it was not a very good lecture, so I'll spare you the rest of it. But the 10th commandment was don't cheat. And you can't really cheat, right? Because you can cheat if your goal is to get a paper or a degree or a grant, right? But if your goal is to cure a disease, you can't cheat, right? And none of it works, right? You cannot cheat biology. So if you keep the motivations right, this will never happen.
Dr. Schrag, would you like to take questions from the audience?
Yes.
So if people are doubting the beta amyloid hypothesis, are there any new ones that
Yeah, you know, there's a surprising number of good ideas in this field, and they've been there for quite a long time. I think that the structure has been that they've been underfunded, and so they're running behind, they're lagging behind. I think the idea, there's 2, in my mind, good ideas that are ready to move into clinical trials now. One is focusing on the tau protein, tangles. That concept has some of the same problems that the amyloid hypothesis has, but there are some reasons you might still want to look at it. One is that the tau tangles correlate much better with memory function. And so it may be that if you can stop the propagation of this tau process, that you may be able to stop it. There's some reasons why I'm not that optimistic about it, but it's certainly worth looking at. The other thing we're seeing, particularly from genetic studies, but also from neuropath and a number of other sources, is there's some smoldering low levels of inflammation in the brain in Alzheimer's disease. And so this concept of neuroinflammation may be a decent target for treatment. And those 2 ideas are pretty mature at this stage and probably the sorts of things that could move into clinical trials quickly. I think there's better ideas, but I think they're further behind than those 2. So we're very interested in lysosomal function in neurons, trying to understand why you're having these clumps of various proteins form in the first place. And I think, as I showed you, we're very interested in vascular function. Because I think that's how you get these proteins out of the brain and clear them through the perivascular spaces. So we think targeting those things may be promising, but they're probably a little farther out.
What kind of legal ramifications are there in this kind of cheating?
Almost none. But don't do it. No, this is usually handled internally, right? So when you raise a concern like this to the NIH, you would assume a lot more happens than really does. And the processes are pretty opaque and slow. And very, very few of them end up in the legal sphere. There are exceptions when this occurs in the setting of publicly traded companies, or the SEC will eat you alive if you get on the wrong side of these issues. So there are some famous cases of people getting in trouble for adjusting data, but very rarely in academics. And there was one individual, only one that I'm aware of, who went to jail from academics for this kind of fraud. And in that case, there were some other factors. He threatened some of his students in ways that I think made everybody see him as a special case. But for the most part, this is not handled legally, although that may not stay that way.
is it basically just the embarrassment of the politics of the university that's involved, that there's the person in the road, but that is.
Well, so when you file a complaint like this, it's interesting the sequence of events that happen. The NIH generally doesn't do much investigation. They refer that to the Department of Health and Human Services, who's an Office of Research Integrity. And they, in turn, refer the investigation to the university. So most universities, I think all publicly funded universities, have to have a Research Integrity Office to handle these sorts of problems. If there is a finding of research misconduct, some of the funds can return to the NIH or whichever other funder supplied them, including the indirect. So there are these conflicts of interest in place that sort of discourage those kinds of findings. And again, the process tends to be a long and a fairly non-transparent one.
So, you obviously gave us a great example about how this is happening in the niche of Alzheimer's, but I feel that it's a bigger problem. It's in every little niche of science that we're all investigating. And how much do you think the stigma of publish and perish is driving this out of science?
Yeah, no, I'm sure there's some of that, right? I absolutely sure there's some of that. I will say though that one of our mantras in this space is that cheaters cheat, right? I will virtually never file a complaint against one finding, right? Because I assume it's an accident or it was innocent, right? My expectations that you're going to be dealing with serial fabulous when you have somebody doing this sort of thing. And so it's a little bit harder to exonerate it across an entire career, surely not over 20 years worth of work where you constantly are jeopardy of your job. Certainly in some cases, I think those images that I showed you when I was working with Hoffman were a time in his career when he was feeling very vulnerable and he was distracted because he'd gone through a divorce. And there were all sorts of reasons why you might soften that blow and say, okay, this is a human going through some things, until you look at the entire body of things we've seen. This is literally 20 years worth of confabulated data. It makes it a lot harder to explain.
You mentioned it taking almost 9 months to get that paper finally retracted. What's the incentive of the journal to keep up a fraudulent paper?
Yeah, I think that people are very, very worried about their reputation, even journals. And I would say don't be, right? I think we worry way too much about our reputation. We should worry a lot more about our data. Yes, sir.
Seems like there's two trends that are making this problem worse, as far as I can see. One is the growth of science teams that get bigger and bigger all the time.
Yeah.
And number two is AI, and so now you can manipulate your background to look random. And so how would you address those problems going forward?
Yeah, I'm going to answer the first question. I'm worried about the second question. But the good news for those of you worried about AI is we're not dealing with simple stuff terribly well. So I figure when we're good at dealing with the simple stuff, we'll deal with AI. Tell me the first question again.
The growth of the science.
Yeah.
teams. You have so many people in a science team.
Yeah.
That how do you know who is the one? Because everyone may have an incentive, whether it's the graduate student
Yeah.
or what have you.
It's really a problem for co-authors, right? I think that now it's very easy to get trapped here because a lot of times you're working in teams where you don't have the expertise to judge every piece of a complicated study. And we need to be able to do those kinds of studies where not every person on the manuscript can take responsibility for every piece of that. What I'll say to that is there are some risks to doing large collaborative studies, but we need to be able to do them. And I think that if you're caught in that position as a co-author, don't think about it too much. Just retract the thing, right? Like I think that we really wring our hands over this stuff way too much. And that if you're stuck in that position, be part of the solution and not part of the problem. But I think you have to be able to grapple with the kind of science that's actually moving the needle right now. Very, very complicated, multidisciplinary stuff. And I think we just sort of have to damp the torpedoes and move ahead with that.
What do you think? Sort of mainstream media is playing into pressure to publish things that might not necessarily be true.
Mainstream media is not very good at science. I don't think that'll surprise anybody. You know, I think that one of the news articles that came out after this said 16 years of funding in Alzheimer's has been wasted, and I think that's clearly an overstatement. But I will say that if we don't self-regulate, this is what we're going to be up against, right? Is people who are not experts in this field regulating us, right? You're going to end up with politicians who are reading those newspapers, creating science police to control these sorts of problems. And I'm not sure that's the most productive way to solve this problem. I think it may become inevitable, depending on the recalcitrance of our systems, you know, but I think if we don't, as a profession, accept that this is an issue and figure out how we're going to make the process more resilient to it, we are going to have processes put on top of us that we're going to have to cope with. Yes.
We have a question from online. How prevalent do you think, excuse me, how prevalent do you think this research integrity issue is across other disciplines besides medicine?
Yeah, that's a remarkably difficult question to answer, and one that we've actually spent a lot of time thinking about and trying to generate some data for. I think it's probably asymmetric. I think there are areas that are doing better and areas that are doing worse. And I think there are certain domains that are more amenable to forensic analysis. And so those areas we tend to find more. Elizabeth Bick, who is one of my colleagues and probably the best research integrity specialist on the planet, systematically analyzed all the papers published in PLOS, which is one of the mega journals over a period of time. And she found that something like 4% of articles had evidence of image manipulation in one form or another. Not all of those would be as egregious as some of the things that I've shown you, but I think it probably gives you a sense of the order of magnitude. PLOS publishes things from all sorts of different domains and is a pretty good sampling of mid-range quality material. And so something like 1 in 25 papers that you read, right? And so it's not every paper by any means, but it's enough that it shouldn't be ignored. Yes, sir.
It's a little bit disappointing to hear that journal editors don't put more emphasis on this. I'm sort of curious. So I have two questions. One, I'm curious about whether or not you see any differences between journals. Okay? And the second thing I'm trying to reconcile is that there are instances where NIH cares about that. So Duke, right? They have that gigantic $1 million settlement.
Do you know why that happened?
Yeah.
Because somebody sued them.
Right.
But that wasn't the government.
Fair enough.
Right. So I think that part of this is that if we want to handle this internally, we need to handle it, well, because what will happen is the legal system will try to correct it, or the political system will try to correct it. And the payout that Duke had to pay was extraordinary, right? And I don't think that'll be the last time that happens. I'm certain there were lawyers who heard that number loud and clear. And some of the things that we're talking about today absolutely could be addressed this way. In the business sector, the work that I did with Cassava Sciences was of considerable value to short sellers who wanted to see the stock price of this inflated company drop. And Cassava Sciences lost $4 billion when our results were published, right? So there are domains where this is taken seriously, and I think it will start to be taken seriously here, whether we do it or somebody else forces us to do it. But I'll tell you, the journal editors, there's a handful that do take this seriously, but the vast majority, some of them have actually been rather threatening interactions, right? I think that there is an unfounded view of whistleblowers and imaging sleuths as the enemy. And I think it's one of the reasons that I stopped the anonymous work, is because I wanted them to see that I'm not the enemy. I'm not some back alley short seller, right? Like, I'm somebody who's trying to move this field forward, and we all want the same things. But I think, again, sometimes you have to put your neck on the line a little bit to move this field forward.
Have you seen any other types of issues with integrity, or do you think that all basically stems from image manipulation?
No, it's everywhere. No, there's.
What is like one of the more common ones you've seen besides that?
Some areas are harder to find than others. Believe it or not, statisticians, very good statisticians, are starting to pick up patterns in data that are unexpected. It's the same sort of thing. When somebody makes up numbers, they're likely to be not quite random. And so sometimes you'll see repetitions or echoes in charts of data that don't make sense. I have a colleague who I think is just incredibly clever, who works in mouse neurobehavior, right? And she's found that you can look at the pathways that mice swim and see that the difference between one that somebody's drawn with a pen versus one that's been actually picked up from a real live swimming mouse. It's interesting because journals won't respond to her, right? She's absolutely right. She's proved it to a Nazi that this is correct. But journals won't respond to that. They're like, mice can swim whatever path they want, but they don't. And it turns out, in her work, she's been very systematic about this, that when she finds behavioral data problems in these manuscripts, and she goes and she looks at the imaging data, there's problems in the imaging data too. Right, so I think our tools for forensics are a little bit better developed in this area, but I think across the spectrum, people are people.
Yes.
Say to research is failing to use the expected data and
Yeah.
using their funding.
So finding the expected data is one of the most disheartening phrases in all of science, right? Yeah, we have this sometimes. The other one that I really love is, I got negative data, right? I just want you to stop at data. Okay? So when somebody tells you they got negative data, right, it's a perspective problem. I made my whole career on negative data, right? Negative data can be a pretty cool finding, right? If you went into something and you thought something so strongly that you knew what the outcome was going to be and you did it right and really high quality work, and you didn't get the finding you were thinking you were going to get, that's great, right? Tell that story, right? Don't apologize for it. That's brilliant. Figure out why, right? That's elite. And I think that this mindset, almost more than publish or perish, is the problem, right? Because the fact is you do have to publish or you're going to perish, right? That's not going away, right? You got to be productive. But I think we have this mentality that if you don't get the expected result, you're wrong. If you don't get the expected result, you should double check that you're not wrong, right? You should go be careful with it. But it might be a pretty cool result. Now, all my best papers were negative. Yeah. Yes, sir.
Another question online. Do you think these articles directly led to the approval of Aduhelm? And how has this behavior impacted actual patients?
Yeah. So the oligomer hypothesis is a little bit broader than just this one paper. So the people who will criticize the interpretation of what we've done here will typically tell you that there's a lot of other oligomers. They're not all A beta star 56. Most people will acknowledge that the A beta star 56 story is dead. Okay, but none of these drugs specifically target A beta star 56. They target other oligomers. Most of them bind a fairly broad swath of these clumps of protein. But this study remains important because of how it was used, right? It's not just the results of the study, but when you look at the formulation of the idea, right? When people went and wrote those big high profile review articles, this was cited over and over and over again at the core of that argument that we should be targeting oligomers, right? Because oligomers correlate with that, right? That was the whole point of the story. And so I think it was hugely influential for driving the thinking about these drugs, not necessarily for the specific development of the specific drug, but in terms of building the argument that we should be investing so heavily in anti-amyloid therapies. It was absolutely important for that.
Do you know of any legislation right now that's in the works to try and make this more punishable offense or have someone face consequences?
I don't, but I'll tell you, within days of this coming out, the legislators in the Senate asked for testimony on it. A number of senators have sent letters to the Department of Health and Human Services to see what's going to be done about it. So I can tell you they're paying attention. And I think that if you hear it echoing across multiple domains, you can count on it, right? Because ultimately, these are the taxpayers' money and a lot of it. Yes.
When looking at a paper and seeing suspicious images and you reach out to the authors and ask for their original image, how do you know that that's their original image? Not
You don't, right? But it's an image, right? And they're telling you this is the source for that, so you can learn a lot from it, whether it is, right? They claim that it's the original image is all, right? From a forensic standpoint, right? Does that make sense? Yeah. Okay.
Are there any ready-made tools or sort of turnkey solutions that article reviewers who work for journals can use to discern whether something has been manipulated?
There's a few. Many journals use them now. In fact, the one that's used by most journals was built by and marketed by a serial fabulist to help other scientists not get busted. And I can tell you that that's why it was developed, because that's what it says on their website. Okay. These softwares are very good at picking up clone statements, right? So they're basically duplication detectors. So when you look at Western blots where the intensity of a band has been turned up or some other things, it's gonna miss a lot of things, but it's not bad for detecting flood stamps. I would say they're not terrible for screening. You know, when I'm doing a large volume of papers, I find it useful to use one of these softwares, but they don't really work well without expert manual overreading. And they certainly don't exonerate a paper, right? All they are telling you is it's not picking up clone stamps. And even when we feed it known clone stamps, it's picking up 20 or 30%. So they're not wonderfully reliable. They're not bad to screen. Yes.
Yeah, so I'm just wondering, we have a lot of students in here, and you can imagine that some environments will kind of breed this type of behavior. So are there ways that students can be proactive about looking for red flags when they're going to different flags?
Yeah, no, I think that's a really important question. Remember, command number 3 was pick good mentors. What didn't make it into the public article about this story with Sylvain Leslie is that we also analyzed one of his graduate mentors from before he was working with Karen Ash, and that person was also associated with dozens of papers that evidence image manipulation. We see that not uncommon, that you see it propagate across generations of scientists from people who train them. And I think that what can happen is that if you're not instilling this as a value, right, that we do not fool with data, it's very easy to, by little increments, normalize this sort of behavior, right? So I would say one of the red flags is you shouldn't hear the phrase, just make it work, right? Like, nobody should be telling you that, right? If you're doing an experiment and you're getting a result that doesn't conform with what you expect, it may be that there's a problem in your experiment and you should figure that out, right? A lot of times that happens, especially as you're learning to do new things. But if you're right and you've done it well and it's not conforming, that's a great result, right? And the data needs to be allowed to speak for itself. So if you hear the words, just make it work, might be a red flag. I'm sure there's others, right? But I like that one. Sorry, guys. Yes, sir.
So I think that the examples you've given, you know, are a little bit easier to track because you're looking at a single image, comparing it. But often some of the problems with these statistics are lack of stability, right? And so it's a little bit harder.
Yeah.
To do process on.
Yeah.
Comment on that.
Yeah. We've tackled those kinds of problems. They are harder. And I would say that almost always when we analyze case, we're looking at a large body of work. We don't look at one paper. And I think the patterns end up being very informative. Having issues with rigor in a study is not necessarily a research misconduct issue, but it can point to that, right? And there are times where that's been really, really important, right? The lack of rigor of the statistics not being quite right for what's going on. You know, there's grades of how serious, you know, we should take different things. The examples that I showed you were all pretty cut and dry evidence of image manipulation. But there's lots of other things that are not as obvious, right? If somebody puts the same Western blot in a paper twice, could they have just mislabeled a file, right? And what we're doing is complicated. The odds of going through your entire career and never accidentally putting the wrong image in paper, we have to sort of understand that. So not every blip in a paper is a sign of some horrible behavior. The patterns are really, really informative. And there are certain cases where you can really infer motive because of the scientific context or the complexity of what has happened. And so we really focus there. And the statistical pieces are just plain harder. I brought a lot of those cases forward that I thought were rock solid and not gotten a lot of support for them. There's probably a way to do it, but I'm not sure we figured it out yet. Yes, sir.
I have one. So it seems like the psychology of the scientist that embarks on this, you know, path of darkness is sort of perhaps a guiding principle for how we can perform our research practices. And so, you know, the impetus to take that first step can be motive. You can put yourself in the shoes of somebody who makes a bad decision. But then at some point there's this transition from, you know, I made a bad call to, many years of this serial fabulism. And, you and I both having watched, people go down this path, would you say that the, principal reason is that after they took that further step, they were trapped and trying to keep covering it up? Is it that they got greedy and thought, well, there are no consequences to this, or is it see that after, they're just not trained well enough in critical thinking or critical experimental techniques that they can't think of better ways to try to, the data are what the data are, we as humans, we're sort of the ones that have to sell the data for that publication. And is it just that in that third case, they never bothered to learn that, or they never trained it, and then they just became a crutch?
Yeah. I don't know the answer to that. Actually, I think that's been pretty elusive. And I suspect it's because people are complicated and they're not all the same, would be my guess. It's interesting, we were sharing our mutual stories around this, and I think that your experience was one of a lot more hostility. Your Othman was somebody that I really cared about, and I think that the impression is very different. I think that going through this with Othman reminded me of this a little bit, right? He's a very human individual to me. He did some bad things and rather unkind to me since I made it public. Right? Understand. But the fact is, he's a complicated guy, and I don't think that it would fit in a tidy way. It wouldn't shock me to find out that most people were talking about it that way too. So I don't know the answer. I tried very hard to get Othman to sit down with a reporter and talk about how it came to happen. But he decided not to do that, but I think it would have been really helpful to understand how this happens and why it happens. But I will say that I think the solution to our systems, whatever we do structurally, is you guys and this kind of conversation, right? There should not be any great badge of courage to have this conversation, right? This should be an absolutely normal part of doing science, is that we talk about research integrity. I was just on the study section, we were lectured mightily. If you have a concern about research integrity, do not say it publicly, do not put it in your written comments, you bring it privately and you don't read the word anybody, right? It should be the 6th criteria on your grant review, right? Are there any concerns about research integrity, right? It should be right out in the open every single time we do this. It should not be an off-limits topic. That's not a terrible place to leave. Should we leave it there?
These things